Hello, ladies and gentlemen, and welcome to today's live broadcast. Nucleic Acid Purification, Understand the Details and Tackle the Routines. Our webinar will be presented to you by Dr. Tim Schalmark, Global Marketing Manager, Automation and PCR at Eppendorf, Dr. Andreas von Bohl, Head of the Bioanalysis Automation Group, and Dr. Milan Diris, Product Manager, Bioanalysis Department, both of MACHERY-NAGEL. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Eppendorf in cooperation with MACHERY-NAGEL. For more information on our sponsors, please click on the sponsors' logos at the left side of your screen. Now, before we begin, I would like to encourage you to get the most out of today's webinar by submitting as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box on the left and click Send. We will answer as many of your questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on that support tab found at the top right of your presentation window or use that Ask a Question box on your left. Today's presentation is educational and thus offers continuing education credits. Following this presentation, those credits can be obtained by clicking on the tab at the top right and following the process to obtain your credits. Before I pass on the presentation to our speakers, I would like to point out that you can read full biographies on Dr. Tim Schalmark, Dr. Andreas von Bohl, and Dr. Milan Diris in the biography tab at the top of the screen. Gentlemen, the stage is yours.
Thank you, Christy, for this nice introduction. This is Milan Diris from MACHERY-NAGEL here speaking. Actually, we've been wanting to do this seminar together with Eppendorf for quite some time now. And the current situation was kind of the reason why we decided we really need to do this now, because basically at this point we get so many inquiries regarding automated nucleic acid purification that we really struggle to answer all these inquiries and to try to get to every customer. So basically at this point, this webinar addresses two kinds of people. On the one hand, of course, labs that are right now in need for automated nucleic acid purification. Most of them in the current situation, of course, may be related to COVID-19 workflows. And second of all, we must not forget that many people, research scientists, lab technicians, students, and so on, are forced to stay at home. I mean, they might not be able to perform experiments in the lab, but they might be at home and want to use this time to plan new experiments and workflows. So they might be thinking about automated nucleic acid purification as well. So this webinar basically aims to give all of these people some background information and technical guidance when planning or considering automated experiments. So let's look at the agenda for today's webinar. I am going to start with giving you some background information coming first of all from the application side. So regarding or when you have a certain application in mind or a certain experiment planned, there are some basic questions that you should ask yourself before starting the experiment, of course. Then I'm going to talk about the principles of nucleic acid purification. We're going to look at like the technological background for commercial purification kits, magnetic beads versus silicon membrane kits. These are the most prominent ones in the market. We're going to show you the advantages of automated nucleic acid purification. This is something that is then presented by Tim from Eppendorf, and he's also going to introduce you to the Eppimotion platforms. And then from our R&D side, with a lot of hands-on experience, Andreas is going to present some example projects and applications, as well as tips and tricks for obtaining optimal results. So for the first part, I'm going to start with these basics. And as I said, in this part, we're looking at a couple of important questions that you should have an answer to before starting a purification workflow, before establishing a purification workflow in general, regardless of whether it is an automated workflow or if it's a manual workflow. There are a couple of questions that you should have answered before starting these workflows. And number one is you should always look at the kind of sample that you're purifying from. So it actually makes a huge difference for the purification workflow whether you're purifying from cultured mammalian cells or from a hard-to-lyse tissue, whether it is a bacterial culture or a complex sample matrix like a swab or something like this. So always keep in mind that different sample materials require different chemistries regarding lysis and purification. So if you think about bacteria, for example, there are also, of course, differences within bacteria. There are bacteria that are relatively easy to crack open. Gram-negative bacteria like E. coli are usually no problem. But there are also some nasty, hard-to-lyse bacteria like Staphylococcus or something like this, so where we really have to assist the lysis either enzymatically or mechanically. So always be aware of the kind of sample material that you're purifying from. Then, second of all, you should always keep in mind or always consider what kind of nucleic acid are you looking for. Is it genomic DNA? Is it RNA? Is it plasmid DNA? Genomic DNA most often is of least concern because if you have a large enough sample available, it's usually no problem to release the DNA and unpurify it. RNA, on the other hand, is a little bit more delicate. There's also lots of RNA in a cell, also lots of different types of RNA. Most people will be looking at messenger RNA to quantify gene expression levels. But RNA is very susceptible towards degradation. So in ambient temperature or in a non-protected environment, it will rapidly degrade. And degraded RNA will then lead to a false qPCR signal. So it will have a huge impact on the results that you obtain. So most RNA purification kits actually have specially designed buffers that make sure that the RNA stays intact. So that's important to keep in mind. And when we look, for example, at plasmid DNA, you should always keep in mind that, of course, plasmid DNA is derived from bacteria usually. Bacteria don't only have plasmid DNA, they also have genomic DNA. And the reason or the method to separate those is the lysis, basically. So plasmid DNA kits usually use an alkaline lysis, which just perforates the membrane enough to release the plasmid content of the cell, and the genomic DNA stays inside the cell remnants and can be sedimented or pelleted by centrifugation. So each and every kit has special buffers, special chemistries to fit your purification or to fit your purification workflow. And last of all, you should always, of course, also design your experiment according to the application that you're using. So what do you need the nucleic acids for? Is it PCR, RT-PCR, a special sequencing workflow, or cloning? Plasmids are also often used for transfections of sensitive cell lines. So you want basically an endotoxin-free plasmid, or even used for gene therapy. And each and every application has its requirements regarding DNA, RNA purity, concentration, quantity, and integrity. So taken together, when you take all these parameters I mentioned into account, the sample, the target molecule, the application, and you're also clear about the requirements regarding like purity, concentration, and quantity of nucleic acid required, then you will be able to identify a suitable method or kit for nucleic acid purification. And that actually brings me to the next chapter, which aims to explain to you the principles of nucleic acid purification, because when I think back to my time in the lab as a student, maybe I wasn't really thinking about what step in the purification protocol does what. What is this buffer used for? What is the purpose of this buffer? Why is it composed like this? It is important to have this background knowledge because you will often run into problems regarding your sample lysis, regarding the purity. And most of the times, if you think a little bit about what is the purpose of each and every step, you're able to solve these problems yourself. And that's often much easier, much less painful to think about in advance. So I want to give you this background knowledge on the principles of nucleic acid purification. So if we look a little bit back into the history, into the late 1970s, people were already using nucleic acids for a wide range of applications. These applications might be simpler or less complex than today's applications, but still, at that time, this was the state of the art. So PCR was not a thing yet, was not invented until the early 80s. Sanger sequencing was not commercially available yet, so all of these things were not there yet in the lab. So what people did is analyzing fragments of DNA that they ran on an agarose gel, like we still do it with PCR fragments today after we did a PCR. And then, of course, they compared these gel band patterns, and they wanted to further analyze single bands out of these gels. So they were looking for a technique to have cut out gel pieces and then repurify the RNA or DNA in that case, repurify the DNA from the gel piece. So that's what Vogelstein and his co-workers did in that paper. So they tested a lot of different buffers that were able to dissolve gel pieces. And with one buffer composition, namely a buffer that contained K-tropic salt in high concentrations, they were indeed able to dissolve the gel. And when they tried to precipitate the DNA again in the solution, the DNA seemed to have vanished. So there was no pellet of DNA. It was not in the solution. It could not have vanished through the air. So where was the DNA? And they found that the DNA was actually bound to the glass. And this was basically, they didn't know about their success at that time, of course, but this was the discovery of silica as a component to isolate or purify nucleic acids. And today, I would say, or in the past 20 years, silica membrane purification kits were very, very successful and made up most of the kits that were used in the market, I think, although we have other kits now coming up again. But these silica membrane kits, they were basically a revolution in the purification of nucleic acids. So how do they work on a chemical level? So everybody knows these mini prep columns, like they are depicted here on the left side. These are basically columns that have a little membrane inside where you basically run your buffers containing the DNA through. And these membranes are basically made out of sheets of silica. And when you look at the chemistry behind this, basically you have a solution, a buffer based on water, a solution that contains the DNA. And when you add K-tropic salt to that buffer, the DNA is stripped of its hydrate shell, and you enable a hydrogen bond between the DNA and the silica membrane. And this is how you can bind the DNA to the column. Then you wash the DNA, and then in the end you can elute the DNA. And this is basically how every silica membrane-based nucleic acid purification kit works. So if we look at the procedure of such a nucleic acid purification kit, it's more or less always the same. You start off with a lysis, which usually includes, of course, your sample, a lysis buffer, and sometimes also enzymes like proteinase K to dissolve your sample in the lysis buffer. Then you usually add another buffer, the binding buffer, which adjusts the conditions in a way that enables binding of nucleic acids to the silica membrane. Then you run this solution through your spin column by centrifuging it, and the DNA is bound to the silica membrane. And then you have usually 1, 2, or 3 washing steps, depending on the type of sample, depending on the purity you need. And in the end, you elute your DNA from the column with a tris buffered solution or water. And then you can use your DNA for whatever downstream application you have, whether it is cloning, PCR, or an NGS library prep, for example. Today we have these silica columns in lots of different shapes and formats. The most popular one is, of course, the mini prep. I think everybody has done plasmid mini prep or a gel or PCR purification mini prep in his life today. But there are, of course, other formats going on from meaty over maxi preps, where you can really put large amounts of cells or tissues as input material. And of course, when it comes to automation, because this is also a webinar that is aimed for people interested in automation, we have these high throughput formats where you can purify from 8 or even 96 samples at the same time. What came up in the past years, and which is getting more and more popular, are magnetic beads. So although, of course, they don't use silica, they are based on a very similar principle, which is binding of nucleic acids to the beads in presence of a K-tropic salt. So the magnetic beads have an iron core which is coated, in that case with a carboxy group, and in the presence of K-tropic salt we have like a very similar mechanism to the silica membrane. The K-tropic salt strips the DNA of its hydrate shell and enables a hydrogen bond between the DNA molecule and the coated magnetic particle. And the purification workflow is, from a chemical perspective, very similar, but from a practical perspective, it's quite different, because you don't process a magnetic bead-based kit in a centrifuge, or you don't process it with vacuum, but as the name suggests, you perform a magnetic separation. So you have a lysate or a lysate where you add magnetic beads and binding buffer, and then the binding of DNA to the beads is enabled, and then you do a magnetic separation by holding your vessel against the magnet. So the magnetic particles are collected at one end of the tube, and then you can switch the buffer solutions and in the end elute your DNA from the magnetic beads. And this is basically, how these kits work. I explained to you like how a silica membrane kit works, how a magnetic bead-based kit works, and since this is a webinar targeted also for customers or lab technicians and scientists interested in automation, we're like displaying the pros and cons of these different technologies here, and later on, especially in the last part, we will see from a practical perspective like what are the advantages and disadvantages of silica versus magnetic bead kits in different applications. And now I think I will hand over to Tim, who is going to jump a little bit into the automation or the process of automation in nucleic acid purification. And okay.
Yeah, thank you, Milan, for this fantastic presentation. I'm going to take over from here, and in the next 15 minutes or so, I'm going to give you a little insight on how to automate nucleic acid purification. So basically, how to get those principles that Milan described previously into an automation background. But before we get to the dirty details, I would like to take you a step back and basically discuss why are you automating a workflow anyhow? So what are the advantages of automation? And for my motivation, I have to say that I took inspiration from a presentation given by Bob Ganser from Beam Therapeutics. So Bob, if you are here in this presentation, thank you for this cool inspiration that you've given me. So if we look at different industries and how they evolved, so if you look at those pictures, on top you can see a picture of a car factory in the early 1900s, and at the bottom you can see a biological laboratory in the early 1900s. And in fact, the laboratory at the bottom, this is the lab of a very famous person. So this is Alexander Fleming. So, and over the time, the way that people work and the way that industries work have changed quite a bit. So if we look at a modern car factory and a modern lab to our right, we can see some drastic changes. So what you can see is that in a modern car factory, a lot of the processes, they have been automated. They have been taken over by robots. Whereas if you look at the bottom and if you take a closer look at the laboratory nowadays, you can see that honestly not so much has changed in this 120 years. And why is that? So the first question is, why did the car producing industries jump to automation, to automated processes? Yes, of course, to produce more cars, but that is most likely not the only reason. There are also other things that have to be considered, which is the standardization of a process and to have equal quality of the product. And if we take a further look at biological research, then there is a very interesting phenomenon. I think that most of you who are listening to this calls are familiar with a thing called Moore's Law. And this Moore's Law says that calculating a capacity of computers is basically doubling every 2 years. And there is another very interesting statistic, and this is called Eroms Law. It's Moore's Law spelled backwards, and it says that the development of drugs is getting more and more expensive over the years. So you can see here in this diagram that the cost for producing a single drug is rising. So basically, the amount of drugs that you're getting for a certain amount of money is getting lower and lower. And this means in order to be productive and in order to be successful also in research is that you somehow have to increase productivities. And that can obviously be done by technologies. It can be done by a smart approach, sure. But I know that most of you have experienced that in the lab that sometimes it is based on a brute force method that you have to run a lot of experiments to get things to work. And this is also where automation comes into play, because this is where most of people start to think about automation. Because if you want to automate a process, you want to get the following done. You want to increase your throughput. So instead of testing 8 samples, instead of doing 8 experiments, you want to do 80 experiments or something like that. But the throughput alone is not the only driver and the most important driver, at least not from my perspective, to automate a process. And now I will go back and take a closer look at the actual application that we talk about. So looking at a genomics workflow, where the nucleic acid extraction or nucleic acid purification is a staple and is a core method. It is also important to understand that this is not a method that is sitting there in a vacuum. So it is part of a larger workflow. It is part of a research question, because I think no one is doing nucleic acid extractions for fun, just because it's so great to run purification after purification after purification. So if you look at this workflow here, at the beginning there is a primary sample where you extract your nucleic acids, and then you have some sort of downstream processing or some sort of downstream analysis, which can be next generation sequencing. It can also be a traditional PCR or a real-time or qPCR, can be different things. So, and if we look at the workload, if we look at the time that you spend to do certain tasks, and here highlighting the nucleic acid extraction, is that a regular nucleic acid extraction looking at 96 samples will involve roughly 500 pipetting steps. It's 500 pipetting steps where each and every step you can do a mistake. And the hands-on time is about roughly 2 hours. It depends a bit on the technology. It depends a bit on your equipment, but if you do it manually, that's a good estimate. If we look at the times for some of the other things here, you can see that the number of pipetting steps and also the hands-on time, it can reach very high levels with more than 1,000 pipetting steps and several hours of hands-on time. So what does it tell us? There are certain tasks in the lab which are very tedious. It means it's a repetitive task that you have to do over and over again. And by repeating this task over and over again, it gets tiring. There is a lot of pipetting steps involved, so it becomes more and more error prone. And by that, it means that the chance of failure is getting higher and higher. So it is likely that there will be a dropout of either single samples or, in the worst case, even of a full assay or experiment if you do everything manually. So if we look at the consequences, is that if you have to do tedious and repetitive tasks, is that your concentration goes down, which obviously increases the risk of errors. I think everybody knows the situation where you're pipetting in the lab, 96 well plate, somebody steps in, asks you a question, you're looking back at your plate, you have no idea where you stopped pipetting. So, and of course, this also increases the number of failed experiments. Another thing is a thing that is related to working in the lab manually with your body itself. So the RSI is a situation where you get problems with your arm, your hand, and your elbow. That means you basically get sick from your work, which can increase the dropout rate, so that you have to take sick leave, you have to stay at home, and you might even get severe long-term problems for that. And of course, and that I think is what most of you are experiencing, is that those repetitive, tedious tasks, they take away time for more intellectual tasks like data analysis or setting up new experiments, thinking about the results from the last experiment. And it is not very satisfying to do that every day. I understand that from time to time it can be a meditative process to do extractions and maybe also do a large number, but on the long run, you won't be very satisfied if you only have to do that. So in sum, it means that if you drop out, if experiments are failing, the cost increases and the productivity gets lower and lower. And this is especially problematic in the sense that more effort needs to be put in to make discoveries. All right, and besides that increase in productivity that you get from automation and throughput, there are some general automation benefits. One, obviously, reproducibility and standardization. It means no matter if you run your pipetting robot, your automated system, today or tomorrow in the morning or in the evening, the performance will always be the same. A robot does not have a bad day, and you can compare results over several operators over several days. You have sometimes, not always, increased accuracy and precision. There is a misconception that a robot will always perform better as a human, which I personally think is not true. If you are an experienced lab technician or a person working very long in the lab, you will be very accurate and precise, maybe even more than a pipetting robot. The question is, will you be as accurate and precise at the 70th well of a 96 well as at the first well? So this is where robots shine. Especially in those days, of course, personal and sample safety is a concern. So if you can give a task to a pipetting robot, that can increase your personal safety, but it can also protect your samples from potential contamination. And another important thing is the documentation and data integrity. So the picture I took here is actually the lab notebook from Graham Bell. I have to admit that my own lab notebook, when I was working in the lab, was not looking that different. Not saying that I am as smart as Graham Bell, just saying that there is not much that has changed within these more than 100 years. There is a big change if you switch to an automated workflow, because you can stop writing up everything manually by hand, because the robot is keeping track of your samples. So sample tracking, sample tracing, reagent tracking and stuff like that is much easier if you work within an automated scenario. So, but now what is actually needed if you want to automate your nucleic acid purification? Well, you need some sort of tool which is handling your liquids. So you need some sort of dispensing tools, ideally covering a volume range which is suitable for nucleic acid purifications. So that is depending on the scale you're purifying, but something between 50 microliter up to 1,000 microliter. That is, I think, is a good range. It can be higher, the volume range that you need if you're looking at bigger preparations, but I think that is a good starting point. What you also need on your liquid handler is some sort of gripping or transporting device, because no matter if you're doing magnetic bead-based extraction or if you're doing a silica-based extraction, you will have to transport either plates to a magnet and back to the magnet, or you have to position stuff onto a vacuum unit or something similar. So this is the thing that you need no matter if you're looking into automating your silica-based workflows or your magnetic bead-based workflows. So if we now take a closer look in what is needed for the two different scenarios that Milan introduced. So if you are automating a magnetic bead-based workflow, you obviously need a magnet. Ideally, this magnet would have a so-called ANSI SLAS format, so a standard plate format, because then it can be easily placed on an automated liquid handler. I'm personally for nucleic acid purification. I'm a friend of rod magnets, as they are shown there, because if working with deep well plates, they will give you a good performance. What you need in addition is some sort, and that has been depicted on top, some sort of mixing module, because in between the purification step, you want to mix your DNA and bead solution in order to get a homogeneous solution to get the best possible extraction results. So if we take a look at a silica-based approach, you would need some sort of a strategy to work with those filter plates. The most common system in the automated world is a so-called vacuum module. So what has happened there is that you put your filter plate with some sort of lid and then by pressure, under pressure, basically the liquid has been sucked through this filter plate. It's been then refilled, for example, with washing buffer or elution buffer, and depending on the collection vessel, you can either collect the waste or the final elute of this workflow. So if we talk about actual systems, so from an Eppendorf perspective, and I will not go into much details here, is that we have two very good options, basically three very good options for automating your nucleic acid purification workflow. One is on the low to medium throughput end. It's the so-called Eppimotion 5073. This is well suited for magnetic bead-based kits, and then we have a solution on the higher throughput range, so medium to high throughput range, which is the Eppimotion 5075. You can see on the right-hand side we there have one model which is capable of operating with a vacuum chamber, so it has a built-in vacuum chamber, so you can work with silica-based kits. And we have one version which has a built-in mixer and shaker, and this is the so-called T version, and this can deal with magnetic bead-based kits. One word for throughput, so we were now phrasing that low to high throughput. Of course, the perception of what is a low and high throughput, this can differ very much. So usually it's the best to talk about sample numbers, maybe sample numbers per hour or per day, because that helps in the discussion either with MACHEREY-NAGEL or also with Eppendorf to estimate what would be the best system for you. But now let's take a look at an actual example of how an automated process can look like, and I also made that very brief because I don't want to deep dive into the software. We choose the Nucleomac pathogen kit, which can extract viral DNA and RNA, and I think you're with me that especially in those times, this is a kit which is of high interest. So what you can see on the left-hand side is the so-called protocol at a glance. So it's the first part of the protocol where it describes you what you would do manually, and I think that is some sort of script that most of you in the lab are following. So if you want to automate that, the first part of this process would be off deck, so it would still be done manually, as this needs to be done under a biosafety cabinet. Of course, the lysis can be the most tricky step in automating such a workflow, because depending on your material, you can either automate it or it might be better to do that manually. And this can be defined by several factors. It can be defined by the kit itself, but it's mostly defined by the starting material. So there is material which can be easily lysed, for example, certain bacteria. There is material which is very hard to lyse, for example, certain plants. In this specific scenario, we are talking about pathogens, about something which is potentially infectious. So in this case, I would recommend to do that under a biosafety cabinet, and then move on to the robot once you know everything has been lysed and safe. So for the second part of the protocol, so everything starting for the binding process, as well as the washing steps, which might be the most tedious ones. What we did is basically we translated the steps in this protocol into program steps on the robot. So you can see it on the right-hand side, and they are human readable. So the software that we are using is very easy. It's drag and drop based. You can learn it very quickly. There are several options. So obviously you can program the Eppendorf robots yourself if you're interested. It's not incredibly hard, so it's something that you can learn quite fast. But we also have a lot of pre-programmed methods ready. We have a vendor and customer qualified methods ready for you, so that that you can use. And we also have services that help you set up the robot and the protocol itself. So how does such a protocol perform? And here are some performance data, both for this particular kit, both for DNA on top and the RNA on the bottom. So what has been done there is that we were using the Nucleic pathogen kit on an Eppendorf 5075 with samples that have been spiked either with bacteriophage DNA or bacteriophage RNA. And then we evaluated the outcome of this purification by a real-time PCR. So it was a reverse transcriptase real-time PCR for the RNA. And as you can see, we were using serial dilutions. And so with diluting our RNA, you can see that we always have, and these are the blue bars, that we have a very good yield, and that the CT value, respectively, the yield is linear to the dilution, so that that shows that the robot is performing very well there. So this is an example for a fully or nearly fully automated process, the Nucleic pathogen kit. I also would like to draw your attention very quickly to something else, which is what we phrase as partial automation. So what you can see here, back to the car industry, is people working with exoskeletons, and it's helping them to do the most tedious or the most tiresome tasks. And this is also something which is possible. So there are very small automation solutions which can be either working like a large pipette, or this can be very small robots where you don't automate the full process, but maybe the most tiresome steps or the most complicated steps. A good example in hindsight of nucleic acid purification would be something like the washing of the plates, adding washing buffer, or also the whole bead cleanup. So I personally would always consider automation if a task is very repetitive and or tedious, if it is very error prone, if it requires a lot of precision, if manual reproducibility is poor, and if samples or operators need to be protected. And of course, if documentation is problematic, if you automate documentation is standardized and processes can be streamlined. And this is these are facts which are true regardless of the throughput. So this is something which is even helpful if you are just doing 8, 16, 24 samples. There are small automation systems out there which can fit virtually every lab bench. And this is, I think, which is very important. So choose the right system for your task. Don't always go for the biggest available option. Look what is really helping you. So you are smart working in the lab, so it's good to have a smart automation at your side helping you to do your daily work. And with that, I want to hand over to Andreas, who will tell us a little bit more on the automation options on MACHERY-NAGEL kits on the Eppendorf itself.
Okay, thank you, Tim, for handing over. Yeah, as Tim already mentioned, we will dig a little bit deeper into the different options of MACHERY-NAGEL kits on Eppendorf systems. Just as Milan has already told you in the beginning, there are two main technologies which can be applied on Eppendorf systems from MACHERY-NAGEL. On the left side, we have the magnetic bead-based purification kits, and on the right side, we have silica membrane-based purification kits, and these two technologies can be implemented on these platforms. Just to show you a brief overview, which kits are already validated or available as validated scripts on the Eppendorf systems. On the left side, you can see a brief depiction about all our Nucleic kits, meaning magnetic bead-based kits, which have been automated successfully on the Eppendorf 5075. You can see it's a very diverse range from DNA extraction from tissue or plant samples over RNA extraction, DNA extraction from food samples, or even, of course, nowadays a very hot topic, viral RNA isolation from clinical samples or from medical samples. And on the right side, we have the silica membrane-based technologies available in two different formats. Of course, on the one hand, the 96 well plates, and on the other hand, we offer a smaller, more flexible format, which is called the 8 well strips. And as you can see, the different kits which have been automated range also about a very diverse set of samples, such as PCR cleanup, plasmid purification, but also other DNA species, such as cfDNA, or even at the bottom, recombinant protein purification in a 96 well format. So in total, around 90 to 95% of the MN high throughput kits have been automated on the Eppimotion system so far. Of course, one of the most frequently asked questions we get is what is the advantage or disadvantage about one technology against each other? So what is the advantage of magnetic bead-based technology against silica membrane-based technology, especially in terms of automation? Milan did already a brief introduction to this in the beginning, and now I want to focus on two kits which have been automated on Eppimotion systems with different technologies, but which tackle the same application, basically the isolation of viral RNA from medical samples. And as you can see on the left, the main advantage of the magnetic bead-based purification is the flexibility. So you can easily adjust your workflow to sample numbers such as maybe 7 samples in one run, or 75, or 48, or 96. So you can really vary the sample number very easily from run to run. With the silica membrane-based technology on the right side, it's more practical to do multiples of 8, because in the end, these kits are processed via vacuum. So you have to seal the unused wells, otherwise you lose the vacuum pressure for the processing. So thereby you are more fixed to a multiple of 8 sample number, or even always 96 samples. On the other hand, the disadvantage of, or the slight disadvantage of the magnetic bead-based procedures are that they always take longer, because you always need to aspirate and dispense more buffers in comparison to the silica membrane-based kits. So you can see on the left 130 minutes approximately for 96 samples with the magnetic bead-based purification kit, and on the other hand only 90 minutes with a vacuum-based, so silica membrane-based purification kit. The advantage of the silica membrane-based kit is clearly also the tip consumption. So the tip consumption is, of course, lower in comparison to the magnetic bead-based kit. On the other hand, the magnetic bead-based kit has a lower dead volume or a lower elution volume, so in comparison to the silica membrane kit. So you can see in the end, it's not that one technology is superior about the other. It's more what is your key application and what are you looking for, which you have to consider for your optimal choosing the optimal technology in the end. Just if you're interested in to dig deeper into specific applications, Eppendorf and MN together have recently published a series of application notes targeting distinct applications, for example, DNA extraction from FFPE samples, RNA extraction, or on the smaller platform for the 5073 isolation, viral RNA, DNA isolation from medical samples, DNA isolation from tissue samples, or DNA isolation from blood samples. So these application notes are all available on the home pages, and please, of course, feel free if you have any questions to come ask them in the end of the session, or to contact us separately. In the next part, I just wanted to focus briefly about 3 exemplary projects and applications Eppendorf and MN have done together in the past. The first project is a medical research project on the 5073 platform, so on the smaller platform for a lower throughput with 24 samples per run. The main application of this lab was to isolate viral and DNA and RNA from swab samples, and then in the next step subsequently subject them to qPCR or qRT-PCR. So basically the main task, the first step of the workflow was a nucleic acid purification using a kit of MACHERY-NAGEL, in this case the nucleic pathogen kit, and then in the second step, also on the same instrument, the qPCR setup was also set up. So this shows that the instruments are not only for nucleic acid purification, but they can also be used for other applications, showing the versatility of these methods, of these systems. The next project I wanted to briefly describe is also a medical research project about cancer profiling. The main application was DNA extraction from FFPE samples. Those of you know or who are working with FFPE samples know how tedious these sample preparation can be. Honestly, I have to say this was one of the most complex nucleic acid purification workflows we have established on automation platforms so far. The reason for this is that with FFPE samples, you first have to deparaffinize the sample, then you have to decross-link, and then you can start with the lysis and subsequently with the nucleic acid extraction. So you have a lot of pre-treatment steps which are usually done manually. And our aim was to automate these steps as good as we can. And in the end, we had a hands-free deparaffinization and purification workflow with a maximum degree of automation. We had only one manual intervention with to a very distinct time point. So you can really put the Glock on 2 hours and you walk to the system, do some manual pipetting, 3 steps, and that's it, and then you press start again. So it was really a minimum of hands-on time. And in the end, what was really nice, the performance was equal to the manual preparation. So in the end, the customer was very happy that this tedious work and very time-consuming and also time-consuming and complicated work could be now automated on the Eppimotion systems. And coming to the last project, one of my favorite projects, honestly, I have to say, I did with Tim together a few years ago in the field of applied testings. This was a national institute, National Forest Institute in Europe, and their aim was the investigation of illegal wood chopping. So basically, they were investigating the they were checking the sawmill waste as well as the remains of cutted trees in national parks, and we're comparing them by DNA sequencing. So basically, it's a little bit like forest forensics or tree forensics. And the main challenge of this was that, of course, we had a kit, the Nucleospin 96 plant kit for this purpose, but as you can imagine, wood or sawmill waste is not the best sample type to extract DNA out of it. So you have a very hard sample type and you have a very low amount of DNA. So, and additionally, this institute also wanted to check for phytopathogens. So in the end, they had a lot of different applications, and in the end, we had to do some adaptations on the method, on the system, but now it's running freely. So although if the kit doesn't say this is specifically for this sample material or this application, there's always something the R&D can check if we can make your application happen in the end. And with that, I wanted to come to the next topic, which will be a little bit the tips and tricks for automated nucleic acid purification. So the session before was more specific to MACHERY-NAGEL kits, and now it's a more general recommendation if you're going for automation for nucleic acid purification. So Tim already spoke a little bit about it. So what points to consider for automation? Of course, there are a lot of different points one should consider before automating. The main points from a technical perspective is always, what is your throughput? So do you have a high throughput, a medium, or low throughput? And of course, what are your application requirements? Maybe you don't only want to automate nucleic acid purification, but you also want to automate other workflows. Maybe you want to do limiting dilutions or sample dilutions on the platform. Maybe you can do the qPCR setup or NGS workflows on the platform. So the versatility of the platform, this is something you should always consider if you're going into the automation. And of course, as the next part, it's always the budget, which is one key factor if you are going for automation. And then in the other part is the technology. So do you want to use magnetic beads Do you want to use silica membrane? So, and of course, does the performance meet in the end with your expectations? And another point which I highlighted here is the service and support point, which you should consider already in the beginning. This is not meaning the service and support from like Eppendorf or MACHEREY-NAGEL. It's more into who is using in the end the platform. So especially in universities, you buy a platform, maybe the PhD student leaves after 3 years because he has finished. So how can the next person easily work with these platforms? So the ease of use for platforms, for automation platforms such as the Eppimotion system is of course a big key factor one should consider. So now I have two examples of the most common troubleshootings we see from the R&D side or from the application side quite often in nucleic acid purification kits. So the first example is the inhibition of downstream analytics due to wash buffer carryover. So basically the symptom is a qPCR or PCR inhibition. And the reason for that is in 90% of the cases that you have a carryover of the wash buffer or of ethanol into the elevate in the end. And this ethanol inhibits then the downstream analytics. So the symptom is always qPCR inhibition, and you can diagnose it very easily if you check the residual volume after each aspiration step on the platform. And to do the volume which should be left in the plate should be less than 5 to 10 microliters. So of course, if it's 0 microliters, that is very optimal, but under normal conditions, it's something between 5 to 10 microliters. You can easily check this by stopping the instrument after the aspiration of the supernatant, taking out the plate, measuring it by a pipetting, and then thereby you can see if the aspiration is efficient as you want it to be. If you see that the residual volume is higher than this amount, then you can start with the troubleshooting. So the first part would be adjust the aspiration height by surface teaching. This is a very simple task which can be done by yourself on the Eppimotion systems. If this does not help or only helps a little bit, then you can increase the aspiration volume. So if you have aspirated before 600 microliters, you then aspirate 650 microliters. And of course, of a third solution, you can prolong the drying step to evaporate the residual ethanol. So these three tips should always help you in combination or as a single task to increase got rid of your qPCR inhibition or to increase your performance. And one of the other most common troubleshooting parts, especially with the magnetic beads, is the low purity or low purity due to insufficient resuspension of the magnetic beads. During magnetic bead-based processing, the resuspension of the magnetic beads in the complete wash buffer is really the key for a good performance. If the beads stay at the bottom during mixing or shaking, then the performance will also be not optimal. You can easily see this because the plates are more or less transparent, so you have a good resuspension when the plate comes off from the thermoshaker, and you can see the beads are distributed evenly in the liquid. So that's a very good sign for a good resuspension. We have a bad resuspension if the beads concentrate at the bottom and the supernatant of the buffers is still clear. So then the shaking was not enough. How to tackle then these symptoms is, first, of course, you can increase the shaking speed. Be careful, do not increase it too much, because otherwise you risk spilling the buffers by the shaking. Another point is you can alternate the directions, so you can change between clockwise and counterclockwise, like in an interval of 1 minute or so. This helps to loosen the bead palette. The other part is what we always recommend is if you aspirate the supernatant after, you can see it in the picture, so you have the magnetic separation, in the middle you have the removal of the supernatant, and then you add the washing buffer, and then you put it on the shaker. You can alter the order or the order of these steps. Basically, you aspirate the supernatant, and then you place the empty plate on the shaker, shake it a little bit, like 30 seconds, then move it back, and then add the wash buffer. Thereby, because you shake the beads without any buffer, you're already loosening up the bead palette, and this helps then with a complete resuspension. And of course, if nothing of these helps, you can still implement a pipette mixing step. As a closing remark, I just want to say usually the mixing efficiency is very dependent on the orbit of the shaker. So if you have a shaker with a good orbit, with a high orbit, then you don't run into these problems. Okay, and with this, I wanted to come to an end and would like to hand over to my colleague Milan.
Thank you, Andreas, for this very valuable hands-on experience. I think this is very valuable information for the audience because it comes really from the application R&D side. And thank you also, Tim, for explaining the advantages of automation in general and of the Eppimotion platforms in particular. We are now at the end of our presentation. We hope that this presentation was useful for you as our audience. We will take questions from you. We have time for a couple of questions. We will not be able to answer all the questions, I think. And yeah, I just wanted already to say thank you to all of you for your attention.
Thank you, gentlemen, for your informative presentation. Let's jump right into that live Q&A portion of our webinar. Now, we have so many questions that have already come in, but if you do have additional questions you would like to ask, just click on that Ask a Question box located at the far left of your screen, and we will answer as many of your questions as we have time for. Okay, I do just want to dive in with this first question. It's come in multiple times. Can we get a copy of these slides?
Yeah, I can answer that. This is Tim speaking. So we will make a copy of those slides available for you. We might have to take out one or another slide or have to adjust a couple of things, but we will make a PDF version available for you, and they will be available via LabRoots as well, if I'm not completely mistaken, Christy, right?
Yes, that is correct. LabRoots will send out an email once we have that downloadable PDF of these slides. We'll send you an email and you can come back into the studio and download those. Okay, let's jump in and get a couple of these questions answered. But I do want to let our audience know that those questions we're unable to answer live today, and any of those that come in during the on-demand period, they will be addressed via the email address you provided at the time of registration. Okay, let's start with this question. And Milan, let's go with you on this question. How can you protect RNA?
Yeah, okay, that's a good question, actually. I think this came up because I mentioned that RNA is much less stable than DNA, and that it is often a problem in workflows involving RNA that degradation is happening. So there are two options to protect RNA right from the start when you still have your sample. So the one option would be, and the best option is to purify your RNA as soon as possible from the sample. So you don't even let the sample sit anywhere for a long time, but you just directly add your lysis buffer and start the purification process. Usually all lysis buffers from RNA kits are designed in a way that the RNA is protected in the buffer once the sample is lysed. And some kits also require the addition of a reducing agent like beta-mercaptoethanol or something like that to further inhibit RNase activity. The other option would be to put your sample into an RNA stabilization reagent. There are several of those commercially available, also from MACHERY-NAGEL. So if you're interested in that, you can also go to our website and just search for Nucleic Protect. It is basically a reagent, and there are, as I said, several of these in the market, not only from our company, a reagent that protects, that infiltrates the sample and thereby protects the RNA from degradation. So these would be your options here.
Thank you, Milan. Andreas, let's come to you for this next question. Do we need any extra equipment for magnetic bead-based nucleic acid purification?
So yeah, so basically what you need is a magnetic separator, which is also available from diverse set of commercial distributors or manufacturers. And then, of course, you need, for the automation, it is recommended to have a thermo shaking device, which is usually equipped on a couple of platforms from Eppendorf, and you need a gripping device to move the plates from the magnetic separator to the shaking device. Of course, you can also do this manually. Then you only need a magnetic separator and a pipette.
Thank you. And Andreas, let's stay with you for this question as well. How can we avoid foaming in nucleic acid purification using silica?
Yeah, so the foaming is a typical issue which comes up with detergent containing buffers during vacuum processing. So what we have established is that in our kit there's a certain wash buffer with a high concentration of ethanol, but you also can use basically 80% ethanol, and we just perform an overlay step. So basically, before applying the vacuum, you just add 150 microliter of 80% ethanol on top of the sample, and then you apply vacuum, and thereby you get rid of all the foaming.
Thank you. Now, Tim, you mentioned partial automation. Does this mean that the system you have is modular and can be adopted step by step, or would that be incrementally?
Yeah, a very good question. So what I mean with partial automation here is that only parts of the entire workflow are automated. So you can distinguish basically between the automation of the full workflow if we take the journey from the sample to the analysis at the end, which can be of different sort. If I refer to partial automation, it's more that within this workflow, there will always be steps which are very tedious, which are very hard, which are very repetitive. So if I think about magnetic beads, for example, there will be a lot of washing steps involved. And it might be tricky to handle, especially plates in that background manually. So it might be useful to just automate that one step. That would then obviously be not a full automation, and you would need an operator working side by side with this automation option. But it is an improvement over a full manual process, and the advantage is that often only very small systems or very small tools are needed to get this partial automation up and running. I hope that answered the question.
Thank you, Tim. And we have time for one final question, and I'm going to go ahead and have you answer this question as well. How do we handle steps like centrifugation during automation when we choose to use silica columns for NA purification?
Yeah, also a very good question. It's actually a question that we get a lot, because if you're doing nucleic acid purification manually, I would guess most of you are doing that with the small spin columns. If you switch to automation, I have to say that most of the time you cannot hold on to your spin columns, but you can usually hold on to the same chemistry and the same process. So in this scenario, the spin columns are replaced by a plate format. It's simply a 96 well plate with the silica placed in this plate. So it's like 96 spin columns connected. And this is then not handled with a centrifuge, but in most of the cases with a vacuum chamber. So instead of using gravity to push your liquids through those filter plates, like with a spin column, you would place it on a vacuum manifold where pressure is used to suck the liquid through those plates. So it's the exact same procedure. It's usually also the same chemistry. It's just a different format of the consumables that you're using. And I think I have to add here at that point that a very large automation solution often have or can have a built-in centrifuge, but then we are talking really about usually a very big system.
Thank you, Tim. And thank you, Andreas and Milan, for this informative Q&A and your informative presentations. Do you have any final comments for our audience?
Yes, I would just like to highlight again that in case you have any further questions, you can go to our websites, which are displayed here. So either the MACHERY-NAGEL website or the Eppendorf website, and contact our technical support at any time with any problem you have related to the topic of the webinar. We will try to answer all of the questions that came in throughout the webinar and get back to you regarding these questions. Thank you again for your attendance.
Yeah, and also from my side, big thank you for your attendance, and also a big thank you for your fantastic questions. I think there were more than 100 questions were pouring in here, and obviously we cannot answer all of those questions live. But as Milan said, we will answer all of those questions via email. Please understand that due to the sheer number of questions you asked, that this might take a little while, but we will definitely get back to you with an answer to your question.
Yeah. Also from my side, thank you for your patience and thank you for your attention, and of course have a nice weekend as well.
Thank you again to Dr. Tim Schammertz, Dr. Andreas von Buhl, and Dr. Milan Diris for their time today and for their important research. We'd also like to thank LabRoots and our sponsor, Eppendorf, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Also be on the lookout for that extra email coming to let you know when you can download a PDF of our PowerPoint. That's all for now. We hope to see you again soon. Have a great day.